Hi, today we are going to kind of put all the pieces together. So, you either just finished testing over the different parts of some of the chemistry of biology and some details into DNA and RNA, nucleic acids, and proteins, or you're about to test. Either way, I'm going to try to connect it all together with a topic that we call gene regulation. Okay. So, one thing that you need to know in order to answer this, come get the lid off my marker. One thing you need to know in order to really understand how DNA make proteins is you need to understand that even though you have a lot of cells in your body, and we're talking trillions of cells in your body, and we call those body cells, the ones that make you up, we call those somatic cells. Even though you have a lot of those in your body, and they all have the same DNA code, they don't all code for all of the DNA. So they don't make all the proteins that the DNA has. And so in each cell in the human body, we have 23 pairs of chromosomes. Okay, so there's 23 pairs of chromosomes in each cell of human. This is for humans. Okay, so that makes a total of 46 chromosomes. Each of those 23 pairs, so you'll have 2 of chromosome number 1, 1 from your mom, 1 from your dad, 2 of chromosome number 2, 1 from your biological mom, 1 from your biological dad, and so on and so forth. And each of those, chromosome 1, 2, 3, 4, 5, 6, all those pairs, has certain traits that are carried on it, certain genes that are on that particular chromosome. And each of your cells inside of the nucleus of your somatic cells, your body cells, have the same 23 pairs of chromosomes. They have all of the genetic material. However, you don't need all the genetic material in every cell in your body. So cells are what we call differentiated. That means they're different. They have a different job. So differentiated cells have a different job depending on what they do. They're different. You can kind of see the different in the beginning. That's an E, by the way. So the job they do is specific to what kind of cell they are. So each kind of cell that you have is different from other kinds of cells. Each of the differentiated cells do not need the same proteins produced that other differently differentiated cells do. So let's take, for example, the cells that are in your eyes. Okay. You have cells in your eyes that make up your eyeball and all the nerves and blood vessels and everything connected to your eyes. Those cells only need the DNA, they only need the genes that code for things to do with the eye. They only need those things. They don't need other genes to be expressed, is what we call it, like producing their protein. We call it gene expression. So expressing the gene that's on the DNA. They don't need, say, you have something in your stomach that's called gastric juices, pepsin. It's an enzyme, and it's very, very acidic, and it dissolves stuff. Your cells that line your stomach, actually there's cells in your liver, and your gallbladder helps to hold it, and cells in your stomach that make this enzyme that's extremely acidic, and it burns through the bonds that are in your food to break it up and so that you can digest it into nutrients. Can you imagine if you had the gene for pepsin, that enzyme that dissolves things? Can you imagine having that expressed in your eye? Like, if that gene, the gene for pepsin, is included in the nucleus of the cells of your eye, but you certainly do not want that expressed. That would be so bad. It would just basically dissolve your eyes because the enzyme is so acidic. And so you can just think that it's absolutely obvious that all of the genes we have will not be expressed in every cell in our body. We talked about lactase and how it breaks down lactose. Well, okay, lactase, first of all, is not going to be produced anywhere except in your digestive system. And on top of that, it's only produced when it's needed. If you never ever had lactose in your diet, then you wouldn't produce lactase, because lactase is only expressed, that enzyme is only expressed when lactose is present. There's no other need to have it. And so you're just wasting energy. Every time a cell creates a protein from DNA, so DNA makes the mRNA goes to the ribosome to build the protein, it requires energy. Cells are very, very careful about the energy they use. You don't want to just waste energy, because if you will need it for something else. So I'm going to talk a little bit today about just a quick topic of gene regulation. Now, this is not something we are testing on like a test in class. This is something that is a whole unit of AP biology. And so I really feel like it's important to introduce it to you before if you're pre-AP. And so gene regulation is basically the cues. It's the cues that cause transcription to begin. So there's got to be something that actually says, hey, we need this gene. It's time to produce it. And so there is. And so we're going to only deal with one particular example, because you don't need the details of it. You just need to know that it happens. So we're going to deal with something called operons. And operons are typically found in like bacteria and things like that. We have a few operons. The more advanced organisms do, but typically this is what we think of as a bacteria way of regulating genes. So operons, if you have, like, let's just say you have some DNA. Okay, so here's your DNA, and there's the little gene pairs thingies. So you've got those. And you don't need the whole segment coded, right? Well, I mean, you might need this whole segment because it's kind of small. But remember, DNA is very, very long. There's, remember from the little poster on the board in my classroom that we looked at that said there's millions of base pairs in one chromosome. And so let's just say that there's a segment of DNA. Like, let's just box this in to say this is the segment that we want coded. So this is our target gene. Okay, so and actually this would be a combination. So these are target genes. So we're coding for more than one gene here because that's what an operon does. And operons are different because they actually they'll actually transcribe like 3 mRNA at the same time. It's like a energy saving thing. And like I said, humans don't have very many operons. We have other things that regulate our genes, but this is the most simple type of regulation, okay? So the target gene is right here. Well, in front of that, we have other little segments of DNA that are like, they code for things, okay? So, so this would be like a promoter region. So it's going to promote the gene to actually go, okay? And then these would be the genes. So let's say each one of these little loops is one gene. So for this particular case, we have gene 1 here and gene 2 here and gene 3 here, okay? And so basically what happens then is if the promoter region is available, then RNA polymerase can get with the promoter here, and there's an operator too, but that's why they call it an operon. The operator's back here. So we'll box it in over here. Okay, so the promoter region is where the RNA polymerase will bind. And remember, the polymerase is what actually makes the RNA. Okay, so the RNA polymerase binds here. And then what happens is if the RNA polymerase can bind right here, then it will take off and start transcribing this gene, okay? So basically, if my little marker lid here was the RNA polymerase, there it is, and the promoter's available, it's like, yay, I'm gonna transcribe this. And so remember, then it would open up, like you know how transcription works, it would open up and mRNA would be released and then would be coded, you know, like if that's an A, then the mRNA would have a U, and if on the DNA there's a T, the mRNA would have an A, and you know how that works. So if this is actually coding, then it would like it would make the little RNA, it would make an RNA for this one, it would make an RNA for this one, it would make an RNA for that one. Okay, it would just be like, woo, make one, make one, make one. Okay, so there'd be 3 little mRNAs that come out of that because there's 3 different genes and they each get their own mRNA. Okay, but there is something called a repressor, and a repressor can clamp on here. And so if the repressor is on, then the polymerase cannot bind. So here comes the polymerase, and it's like, oh, where's my gene sequence? I can't find it. I can't find it. Okay. So basically, this is a way of turning off the DNA that you don't need to be transcribed, or if you remove the repressor, turning it on so that the gene will be transcribed. Okay. And that's what an operon does. And so these mRNA, that are these 3 mRNA that come from these 3 genes, they are only transcribed if necessary. So for lac operon, so this is lactase. See if I can write it somewhere you can see it. So for lac operon, so that makes lactase. That's why it's called lac operon. Then it would only, the repressor would be on there unless lactose is present, and then it would remove the repressor. So lactose has a little binding thing that would bind to the repressor, which is also a protein. So it would bind to the protein like you saw in class with the enzyme and the substrate bind. It's like that. It would bind to the protein that is the repressor, and then the RNA polymerase would be able to code for the correct gene to be produced. Okay. And so then, of course, then we know what happens after that, of course, then our mRNA. So our mRNA would then travel to the ribosome. So it leaves the nucleus where it was created in transcription, travels to the little ribosome, which looks like a little flat snowman. So there he is. And that is made of our RNA. Remember that? And it would travel there. The mRNA would go in between there, and then it would be read, and the proteins would be produced. And if it's a lac operon, lactase is produced as one of those. And then also the other 2 are things that go with lactase. So things that allow lactase to enter the lining of the stomach and stuff like that. Okay. And so that's basically what would happen. So the central dogma is where, remember, when transcription creates mRNA and translation then creates the protein. Well, it's not just on all the time. All of those DNA have a signal. Now, some of them are on all the time, but they can be repressed, like they can be turned off. So depending on what you're needing, if it's something that the body typically needs all the time, then that transcription will be on all the time. But if it's something that's only needed sometimes, like lactase is only needed when lactose is there, then it will be off unless it's needed to save energy. Okay, so that's an operon. I hope that's very understandable, and it helps you to also review the parts of protein synthesis. Have a great day, and I'll see you soon.